Hello, my name is Cristina Baciu and I am Scientific Associate in Bath Lab at UHN, Toronto, Canada. I'm very pleased to present today an overview of our project using machine learning for diagnosis of liver graft pathology. Liver transplant recipients are very closely followed during their trajectory after transplant. They can have graft injury of various etiology, resulting in graft cirrhosis over time. Reasons for elevated liver biochemistry in graft injury include T-cell mediated rejection, recurrent or de novo non-alcoholic steatohepatitis or NASH, viral hepatitis, or biliary complications. Liver enzymes have altered levels in all, but the management of specific etiology is different. Currently, liver biopsy is the gold standard for diagnosis, but is invasive, associated with multiple risks, and is often delayed. Several non-invasive markers, such as images or serum biomarker, exist, but most of the time their sensitivity and specificity is low, and in case of serum biomarkers, they cannot distinguish between different stages of NASH, for instance. Also, donor-derived cell-free DNA tells us there is a graft injury, but not the cause. Therefore, there is a significant need for non-invasive biomarkers. A new innovative protocol, CFMDIP-seq, looking at plasma cell-free DNA methylomes, was published by the Carvalho Group and collaborators. And this is a sensitive protocol to analyze the methylome of small quantities of circulating cell-free DNA and is able to detect large-scale DNA methylation changes that are enriched for tumor-specific patterns as shown in this figure. So we aim to develop an ML-based tool leveraging methylation patterns and clinical variables for accurate classification of liver graft pathology. Selected clinical information of the patients in this study are presented. To note, the enzyme levels are significantly higher in the disease group versus control. And of this, liver enzyme, ALT, AST, ALP are the most important. And we also use the indication for transplant for the modeling. Here we present the bioinformatic pipeline applied to CFMDIP-seq raw data, followed by the methylation enrichment and differential methylation analysis with a couple of R packages. When comparing NASH LT patients with controls, which are also liver transplant patients but with normal level of liver enzymes, we identified around 2,700 differential methylated regions with good level of CpG enrichment level as shown on the left panel. With the genes from the genes body, we have performed pathway analysis using IPA. We found that lipid metabolism with lipolysis biological function was activated in NASH patients. We also compared T-cell mediated rejection patients with controls, and we identified around 1,900 hypermethylated and 700 hypomethylated regions. In terms of canonical pathways, IPA identified activation of platelet-derived growth factors in TCMR patients in agreement with previous studies. IPA revealed activation of gene expression as well as cell cycle activation. Transcription and phosphorylation of amino acid were also highly activated. The processes were predicted to be regulated by specific transcription factors as upstream regulators presented on the top of the networks. We then developed a machine learning approach with main contribution by Dr. Sumita Ghosh. With 43 patient samples using methylation data and clinical variables, we generated 101 stratified randomized 70-30 trained test splits. The principal component analysis plot and the heat map with the top 300 differential methylated regions used for the modeling showed fair separation by disease with 49.98 of the variance alone explained by the first principal component. Using multinomial logistic regression with L2 regularization as our classification approach, we independently trained and evaluated the models using the bootstrap datasets. We also evaluated various performance metrics on the test datasets using one versus rest approach. Overall, the bootstrap models achieve high classification rates for control and TCMR with mean true positive rates or TPR 90% and 95% respectively. The models found Nash LT class to be the hardest to classify with mean TPR of 82%. And the table below includes the mean accuracy and other metrics for the 101 computed models in the test set. Feature importance analysis or Shapley analysis identified the top-ranked feature, including several DMRs and a few clinical variables such as ALP and creatinine. So in summary, our machine learning tool that we call Clever LG, leveraging methylation pattern with clinical variables, significantly improves classification of graft pathology over individual modalities. We also find that ALP and creatinine are important contributors in models decision. And overall, Clever LG shows significant promise as a specific non-invasive tool for prediction of liver graft injury. And now I would like to thank DataBat in the center of the image here in red, and lab members, in particular Dr. Ghosh for machine learning analysis, to our sponsor, and to all for your attention.